Good day, I'm Dr. T and welcome to my office. Today I'd like to talk about genetics, but not necessarily in the way you usually think. One thing as chemists that it's kind of our gig, is we take what other sciences take for granted, or things that other sciences take as their foundation, and we dig deeper. So the question is, why is genetics genetics? So a little of a brief overview, stuff that hopefully you're familiar with from a biology course. Okay, so you've got two copies of your DNA. One came from your ma, one came from your pa. Now, it turns out it's not, if I take your DNA out and I can't pull, oh, this is your ma's strand, this is your pa's strand, because shortly after conception, your cells take the two strands and shuffle them, and they keep shuffling. You know, it's not a one-time gig. So now your strands are shuffled, so it's parts of it from your ma, parts of it from your pa, but it's not on the same strand. By the way, this is why different species, their children sometimes are sterile, like a mule. That's because when you shuffle things over, they don't shuffle the same way, and thus you can get a copy that has two copies of one gene and the other copy doesn't have a copy because of where they're placed. That was an aside. Moving back. Okay, so you've got two copies of your gene. Now remember, different versions of a gene are an allele. Okay, so I have two different alleles. Now there's actually a little bit more going on. So I've already talked about producing mRNA, and in that I mentioned a little bit about the promoter. So you've got the part of your DNA that codes for the mRNA that will then code for a protein. Bada boom, bada bang. But you've got more going on. You've got part of the DNA, mostly right before it, but it is scattered out through the DNA, that controls how that mRNA gets used. The region before the mRNA, or the sequence that will be copied into the mRNA, is known as the promoter. The promoter attracts transcription factors. The transcription factors then attract the RNA polymerase. The RNA polymerase then actually does the copying. So effectively, the transcription factors and the promoter area is almost like the operating system of the cell. Okay, so we've got this region. This means that I have a couple of places where mutation could change things. So if I change the promoter region, or any of the control regions that are not necessarily the promoter region, now all of a sudden I could take a protein and turn it off. Just never gets around to getting made. It doesn't actually attract the transcription factors. Alternatively, I could have it attract the wrong transcription factors, or I could make it go nuts, and so it really attracts the transcription factors, and therefore it's produced either at the wrong time or way too much of it. And there are definitely diseases where this is the case. You either don't produce a protein you needed because the promoter region is messed up, or you produce at the wrong time, or you produce way too much of something, etc. Now, those transcription factors are actually fairly important. They interact with kinases, other molecules, and thus function, once again, that kind of computing concept. Additionally, I could have modifications in the part that actually becomes my protein, and so I could have a protein that does not work as well. It's possible that it works better for a particular role, or it just doesn't do anything. Especially if I get, a nonsense mutation or something nasty in there like that. Now, this means, though, that I have two alleles of every particular gene. Now, you're familiar with this idea of dominant and recessive genes. It's actually a little bit more complicated than that. There's things like codominant, etc. Typically speaking, though, when I'm thinking of a dominant gene, I've got an enzyme. And actually, let's think about me. If you zoom in enough, you see I've got brown eyes. Now, I happen to know that I'm heterozygous for brown eyes. My mother has brown eyes. My father had blue. Okay, so if you look at that, I've got two alleles. One codes for brown eyes, one codes for blue eyes. It actually doesn't code for blue eyes, though. It's actually coding for brown eyes, but not succeeding. The native color of your iris is blue. That's why little children who haven't developed enough pigment have blue eyes. You know, the baby blues? There. So, I have two copies. The one from my father... brown eyes that aren't brown, therefore they're blue. His enzymes were not producing the pigment, therefore blue eyes for him. But I have that enzyme which isn't making my eyes brown, and I have a copy from my mother. Her pigment, or her enzymes that would make her eyes brown, do actually make her eyes brown, and thus my eyes are made brown, because I have one working copy and one that doesn't do it. Hence the fact that brown and blue, one is dominant, brown, one is recessive, blue. It's being hidden. Now, in some cases, you do need both alleles in order to make enough of the pigment. In that case, you could think of a red rose, a white rose. The red rose makes the pigment, the white rose does not. And heterozygous, remember, homozygous, two of the same allele, hetero, one of each. The heterozygous, you make some red, but not a lot of red. So what do we call some red? We call that pink. And yay, you have yourself a case where it's not a textbook, dominant versus recessive. Also complicating things is a lot of things end up being not just one enzyme. So some things like pea plants, they're nice. It's one gene, one protein, it does a lot. Whereas, people, it's not one gene, one protein. There's a lot going on, which, has had some interesting effects in human history that I am not going to get into. So with that said, have a wonderful day.